So we have the scientific method. I get it. Okay. I mean, I'm going to fly through parts of this scientific method, because if you've had 101, which I'm sure you've had 101 at some point, or some kind of science class, you've looked at the scientific method. And I mean, this is all the nonsense that I just discussed and talked about there with that classic example of pellagra. You make observations, you formulate a hypothesis, make and construct a study, collect data, analyze the data, and show the results to your peers. And then they can poke holes in all your stuff. And then you do it all over again. Okay. Now, I mean, you can review literature. Okay. You could see what other people have done. And you could take their study and try to copy it and see if you get the same outcomes. Or you could take it and, they didn't take into account a variable, and you're like, okay, I want to take into account this variable and now see what happens. And you get a lot of that in nutrition science, okay, where they'll do the experiment. And it's the great thing about these now, usually at the latter end of a research article, in all science anymore, not just nutrition, they'll put, potential flaws in some of the study, or if there's any biases that they have, like this is a study on milk, and it was shown that milk was the holy trinity for, curing all your delayed onset muscle soreness after exercise. Down in this area where they'll put potential conflicts, they'll be like, this study was funded by the Dairy Association. And then you can take that for what you want. Okay. So they do a lot of great things now in scientific studies that they didn't do way back in my day. And even some of these studies have been flawed dramatically, okay, where they've manipulated data. And there was a huge rigmarole, I think, in JAMA back in late 90s, early 2000s, where there was a lot of research in those articles being presented, and it was not good science at all. It was manipulated data. So I don't know how they came about figuring that out. I don't remember. But the source of dietary recommendations should come with science. Okay? Should come in science. And that's really hard because science is really difficult to read. Nobody picks up a scientific journal and says, I'm just going to chill out and relax today and read, nutrition reviews today. That ain't happening, right? You're going to see whatever floats around on your Instagram, on TikTok, whatever the hell you guys read anymore. Okay? So it's a lot of anecdotal information. Okay? Anecdotal information is not scientific at all. What's anecdotal? Well, you should do keto because I lost 1,000 pounds. Well, let's be more specific. I lost 85 pounds on keto. So you should try keto because I think it'll work for you. That is an anecdote. Okay. That's your personal response. That's your personal report of something that you did. You could say, don't try keto. I didn't lose any weight. I gained weight. Okay. Anecdotal information. And a lot of nutrition floating around out there is anecdotal. Okay. We want to try to use a lot of scientific research. And that's why I have like 1,000,000,001 clicks, links, PubMed ID article numbers, all this fun stuff scattered throughout to try to show you some of the current research that's out there. The bad thing about this version of the textbook that you guys have, it is obsolete. I think you guys have version 2. We're doing version 3 in the fall. So there are some links in the book. Some of them are just really not very good. And I've put a whole bunch more in here. So when you get a clickable link, click on it. Go down the rabbit hole. Read around. Look at some stuff. Because it's pretty fascinating information and research being done out there. What do we usually do experiments on? Animals. Okay. Experiment, what is an experiment to begin with? It's right there, just a way of testing a hypothesis. Okay. We use animals. We primarily use mice and rats. They very genetically similar to humans, okay? Fruit flies. Fruit flies. We don't necessarily do them with research with nutrition, but we do a lot of research with fruit flies because they have the same kind of sexual passing of information from one generation to the next. So that's why we do this research on animals, on genetically similar animals anyway. So when it comes to nutrition science, we do it with mice and rats because they're genetic, very similar to us. They have biological and behavioral factors or characteristics that are very similar to us. So that's why we use mice and rats in that standpoint, from a genetic standpoint. Very convenient. Okay? They're small, if you didn't know. We can keep hundreds of them in a very, very, very small place. And they adapt very well to their surroundings. And that's pretty much like humans. Okay? They reproduce very quickly. Okay, so it's not like us, where if I want to get information on what this does for generations down the road, that's really hard. That's a very, very, very long study. Okay, that's going to be multiple examinees doing this type of experimentation. It takes a really long time. So mice and rats, they reproduce very, very, very quickly. They have a very short lifespan, around 2 to 3 years. And so we can get a lot of information. Relatively inexpensive. They can be bought commercially in very large quantities. So, you know, generally mild tempered. So they're not going to go rabid on you and get crazy like some kind of horror movie that you would watch. We can do, there was a question we can do on primates and whatnot. But again, that's a lot more money involved. They're going to have a much longer lifespan. So again, it's not optimal. Mice and rats are. That's not to say we don't do experimentations on monkeys, on humans, but mice and rats, I can control everything in these creatures' world. Okay, I can knock out some genes, make them live longer, make them shorter in lifespan. Okay, there's a bunch of things that we can do with mice and rats. Okay, that we can't do with humans. I can't lock a human up legally. I can't lock a human up, right? So I can't control every little aspect of this human's life. A mouse? Yeah, I can control everything. When they eat, when they exercise, what they eat, what they exercise, how long they exercise, make sure they're doing the exercise properly. I can make that very rigid for them. I can control that very well. Very difficult to control a human, as we all know. Okay, there are guidelines. Okay, there are a number of guidelines, so it's not like, you know, this is unethical. It's not like you are, you know, cutting off a limb and making them run around and do stuff or anything like that. And if you ever go to a big university where they do a lot of research, you go through this. Okay, you go over the ethical treatment of them, how to do about it, how to maintain an aseptic environment, how to handle them. They're really pretty docile creatures in the grand scheme of things. And surprisingly, they're pretty big, which is weird. There are a vast number of different forms of studies, and we'll look at a couple major ones here. But when it comes to the subjects, we can use in vivo studies or in vitro studies. Okay, and I have the stuff that's underlined, and that's probably the distractor that you would see or the choice that you would see for a multiple choice question on an exam. It's generally why I have stuff underlined anyway. So in vivo looks at the whole organism itself. Okay, and that's what we're primarily looking at. Okay, when it comes to these studies. In vitro, we can do those as well. We'll take a cell culture and, expose it to, a carcinogen, or expose it to, high amounts of a certain type of vitamin, mineral, whatnot. And we can do that as well. Or it can be components of a living organism. Again, it's generally just looking at cells or tissues, whatever, organs or whatnot. Okay. So we can do that as well, but in vivo is going to be the most, because again, it looks at the whole organism itself. To test, obviously, I need two different groups for a successful experiment. I need an experimental group, okay, or the treatment group. One version of the text has experiment. One version of the text has treatment, and I can't remember which one in my head because I've been looking at the third one, so I can't remember. But we have a treatment group or an experimental group. This is the group that receives the treatment. They are the experiment. Okay. And then we have a control group. Control groups do not receive that treatment. Okay. What are they going to get? Anybody know the word? It's a P word.
A placebo.
Yeah, they're going to get that placebo. Okay. They're going to get that sugar pill, generally. And then we can take that, and then we can look, and we can look at the difference between the two groups. The groups that were the treatment group or the experimental group, and the groups that were the control. Okay. And then from that, we can see if whatever we were testing for had a positive or negative effect on it. And there you go. Okay. You got to have a control group. If you don't have a control group, it's really hard to figure out if whatever you were experimenting means anything. And there are a number of studies that, may get thrown out there that may not have a control group. It's designed to be we had a number of patients, let's just say, and we noticed a pattern. And so we went back and started looking at what all of these individuals had. And they may throw together a little bit of a study, just looking at that one thing, not necessarily having a control. Not necessarily a bad thing. We just can't gather any true concrete evidence about whatever was being tested on. Okay. Usually you'll see those, and then this is like the first step into a long-term type of study. And so you do see those out there, but generally they dovetail into an experiment with a control group a lot of times. So you will see those out there. I mentioned it earlier. Okay. We want to control variables. Variables are factors that can change the outcome of a study. And that's why we love to use mice, rats. Okay, because I can control damn near every variable that I can think of. And invariably, there's going to be some variables that pop up that you didn't account for, didn't think about. You'll put that in your scientific paper, and then hopefully your next study, either you or some other person, will control that variable. Okay. They got to be controlled because, it can make the study super inaccurate. Okay, super inaccurate. So if you don't control for these variables, and in scientific nutrition, that is a big one. Okay, because sometimes it's hard to control every single variable. That's why research in humans is really difficult. Okay? Really difficult. Because when it comes to humans, they are, quote unquote, free living. Okay? They're out there interacting with the world. They're going to be drinking things that may be different from one to the other. One may have coffee with 2 sugars. One may have coffee with 4 sugars. Okay? One may skip a meal. One may eat every meal. And then the meals may not be equated for constant protein throughout the entire day. And they may go high and low on protein content while going high and low with fat and carbohydrate. And you have to keep protein constant throughout with these type of studies. So it's really difficult to do this with humans, all right, because of that. One may drink an extra soda. So there are a number of fantastical different things that make it difficult to do studies with humans. Okay, even though, do we do studies with humans? Hell yeah, we do. We obviously do. It's just very difficult. And so when we're talking about some of the variables when it comes to human research, huge importance. And that needs to stand out in a lot of these studies. It's something that you should be looking for when you start to research, do that nutrition in the news assignment, where you start looking at, something that you read on Men's Health, okay? And you're like, okay, that's pretty interesting. And then you try to go in and you find some scientific research articles on that, and going through and seeing if this article in Men's Health was supported or not supported by the scientific research. So looking at some of those variables will allow you to really figure out if this research article is good or not very good because of that factor of the variables. There's placebo if you didn't know, okay? Given to given to the control group, and that helps us to ensure that the treatment was valid. We are going to have a few different types of studies: cohort studies, observational studies, all sorts of fun stuff, epidemiological studies. When it comes to these, when it comes to human experiments anyway, they are pretty much going to be double blind. In this type of situation here, this is a study where nobody knows. Scientist, the investigator, or the subjects, they have no idea. Okay? This helps us to eliminate or avoid or not get into two major things. One, the placebo effect, okay? And two, experimental bias. And this, we always talk about the placebo effect, okay? But really, this one is just as important. Okay? If you know that an individual is receiving the treatment and not the fake treatment, you are going to interact with them differently. We're human, we just do. Okay? Getting rid of that experimenter bias is really important, because this ensures that really everything is going to be done equally among every single person in the group. Placebo effect here is just when someone reports having positive results even when they are taking the placebo. And when it comes to placebo effect, especially with supplements, this is usually a lot of the case. We'll talk about some supplements later in the semester. The book looks at supplements kind of evil, at least in this version of it. In the third version of the text, it's not as evil, but it's still relatively evil. But when it comes to supplements, there is not a whole lot of rigorous scientific research done on a lot of these products before they get out into the populace. And you see a lot of placebo effect where they take it and they start to feel like it's having an effect, even though it may not be doing a damn thing. Okay. And when it comes to a lot of supplements, like one supplement I saw, and this always makes me laugh. It was something that was going to help to boost your muscle synthesis and make you lift and do all this stuff better. And once you got down in through past the advertisement and got the actual, then they show you, you know, the front and back of the bottle with everything in it. The primary thing that was in it, the primary ingredient, the first thing labeled on the thing, you know, where it says list of ingredients, the first thing is always the greatest. Tomato sauce. Tomato sauce. Tomato sauce. What the hell? Come on. Tomato sauce ain't doing that. Okay? Tomato sauce is not doing that. When you go through and start reading some of the study, when you get past some of the glitzy graphs and whatnot, it was just looking at, you know, cellular experimentation, and it didn't really do anything. Okay? But it was the placebo effect. Okay? The individual took it, thought that they were feeling better just because they took something before they worked out. And who doesn't take a pre-workout? Does everybody work out? Some of you do. Some of you I can see foreheads and I don't know. Some of you I can see pictures. So do you work out? Okay. Yes. I'm just going to go with the positive here. Do you take a pre-workout? Brett says yes. Okay. And you take it and you feel better. And let's say one morning you don't take it. And now it's in your head. You're like, oh crap, I didn't take my pre-workout. I'm not going to be able to lift 90 pounds anymore. And now you're going to have a negative connotation with it. And now that may set you up for failure in the gym because you always have felt good taking it. That's the placebo effect, ultimately, in a nutshell. Okay. Sometimes some of those pre-workouts, I take a pre-workout. Some of them are filled with a lot of niacin or caffeine. Niacin helps you feel good. I mean, kind of gives you that tingly feeling. And if you have way too much, holy Christmas trees, man. I took one time. I thought like lightning was going to come out of my fingertips. It was insane. But that's a side effect of really, really, really high niacin. Double-blind studies, epidemiological studies. Epidemiological studies. What's an epidemiology? Epidemiology looks at the occurrence, distribution, and the causes of health problems in a population. Okay, this is what we use a lot of in nutrition. This is what we can really utilize to really see if diet has a huge influence on health. And so these are what we are going to be utilizing a lot of. Sometimes they are observational in nature. Sometimes they are a cohort, and we'll talk about those briefly here in a second. Cohorts are kind of the best in my mind. Okay. These studies can be done a couple different ways, like I just explained, but we use these, or an epidemiologist uses these kind of studies. And when they do these studies, they gather information. Okay. The more information we can gather, the better the study. Okay. The more correlations that we can draw upon. Right? And if we can get a causation, yippee skippy. All right, yippee skippy. But it's all about collecting information. And this over here is information. This is health information right here. Height and weight, length in this little individual right here. And again, 20 women in your office. Let's just say you have 100 women in your office. And they all have a kid relatively around the same time. I mean, that's 100 data points of information. 100 is not a whole hell of a lot in the grand scheme of things. Right? I want to get a lot of information. And so we just put this, as physicians, we put this into various different databases. And then if we get hundreds and hundreds, leading to thousands and hundreds of thousands of data points, now we can really start to see what happens if little babushka there doesn't get breastfed properly or isn't eating enough properly. Okay? What that does for his weight. Another piece of information they'll take from that little bitty boy right there, they'll see how big his head's getting. And that correlates to brain growth. So there's a number of data points and, height, weight, how big their head is. Those are kind of the big things that they do when it comes to babies anyway, to see if they're growing properly. All right? So this data is necessary to come up with all of our recommendations that we're going to be talking about in chapter 3. This thing is called the National Health and Nutritional Examination Survey. Why is this important? Because researchers can use all of this data that has been collected from all over the United States to determine the prevalence of the disease, how many people are coming down with it, high blood pressure, let's just say, and then the risk factors for those diseases. What do all of those individuals who have high blood pressure have in common? Is it weight? Is it body fat percentage? Are they smoking? Are they all eating a certain type of food or consuming a large amount of a certain type of nutrient? That's what we can figure out from getting all of that data from a vast number of information of individuals across the nation here. And that's what we do. Right? All of these standards that we're going to be talking about in chapter 3, right? All of them are based upon these surveys that are submitted so we can get an idea of average height, average weight, BMI averages for every single state, the incidence of obesity for states, for regions, for specific ethnic groups. And then we can use that information to target that specific group to make sure that they're getting access to the healthcare that they need to have a wonderful, healthy life. Again, we can do correlation studies, we can do causation studies, okay, when it comes to these. But a cohort is just a group. Okay, cohorts are just a group. So when we get a number of individuals together, like all you guys right now are a cohort. You're all around the same age. When we come to the two types of studies that we're going to be looking at here in a second, observational studies, we will put them into, excuse me, we will put them into groups that are similar, same age. Okay, and no, not all, let's just say 20. Okay, but 20 to 29, that's a group. Males, females, black, white, Asian, Mexican, race, social status, those with large amounts of money, low amounts of money. I have some links to some stuff through the CDC that looks at BMI, and it looks at all of this stuff. It's got it all broken down for age groups from 0 to the grave, okay? 0 to 100. Incidence of body weight, BMI. Breaks it down by sex, males and females. Breaks it down by race. Even more fascinating. And then there is a section in there on access to money. Those who have a large amount of money, those who have a low amount of money, those who have a high degree of education, those who have very low degrees of education, and what that causes for BMI. Fascinating. Okay? Fascinating. And from that type of information, those that are poor tend to have poor health. Why is that? Well, that could be due to poor access to healthcare, affordable healthcare, access to food. In a lot of those regions, there's a lot of food deserts. There's not enough access to real whole food. And so we can try to figure out what's wrong with that specific region in terms of health, try to fix it. There are a number of studies out there, and I'm only going to be talking about observational studies here. There's ecological studies. There's ecological studies would look at a geographical area, compare it to another, and look at incidents of like cancer mortality, and see kind of what that, what the environment is doing there, what's in that environment that's lending that to cause that. There are cross-sectional studies. They look at like, they'll look at a specific type of condition, asthma, respiratory conditions, let's just say, and they'll look at the prevalence of that in that specific region, and try to figure out, what's the cause for that. Sometimes it's manufacturing. There's going to be a lot of times with those type of studies involved in those type of studies. Like there's a certain type of industry that's there. There's going to be a vitamin that we'll talk about later, and it's very mundane, but if you work in a certain industry, if you're a miner, you get exposed to severe toxic amounts of this mineral, and it causes hallucinations. It would be one that you really don't have to worry about finding in your diet in that case. So that's just because of this region is, Pennsylvania, West Virginia, and areas where they do a lot of underground mining a lot. So the only one that we're going to be looking about is observational. It's the only one besides epidemiological. Observational studies, this is what the vast majority of nutrition research is, where we have a group, a cohort, okay, and we can follow this group going forward or looking back upon things, okay? But that's what a cohort is. So if anyone says, oh, well, the cohort is, well, now you know what the hell that means. All right, so case control studies. Case control studies are very quick, very inexpensive, okay? So from that standpoint, they're really easy and cheap to do, okay? That's why they're done. The bad thing, so these would be advantages here. Disadvantage, it's usually a smaller number of individuals, okay? If it's quick and inexpensive, generally that means fewer numbers of fewer numbers of individuals involved. So that would be the one disadvantage. So you would have to do a large number of case control studies to really get a really good idea of what, kind of correlations that you can kind of demonstrate there. Why, if these are so bad because it's disadvantage, a small number, why would you do these? You would do these if you were investigating an outbreak where you need you need a lot of answers really, really, really quickly to help slow the spread of that outbreak. Studying rare diseases where there's only a very small number of individuals that even have this disease or disorder. That would be another kind of situation where you would do these. So not bad. They're just not the best because of the small number. So you can't apply it to a huge populace, okay? I have an example of their study in which an individual with a health condition, heart disease, okay, are matched to persons with similar characteristics, but they don't have it, okay? So we have our controls. These are the individuals that don't have heart disease. high blood pressure, atherosclerosis, heart disease is just kind of a catch-all for all that. And we have the same group of 20-year-olds. We have 20-year-olds who don't have heart disease, and we have 20-year-olds who do have heart disease. Okay. And then we can figure out correlations that way. Okay. They do not demonstrate causation. They do not demonstrate causation. Small number, can't get it. Cohorts. These are really pretty good. Cohort studies are. Why are they really good? Because we can get a very huge amount. Sorry, I hit something here. We can get a huge number of individuals for this thing. And we can follow them, as you can kind of see down there from the examples on that picture there. You can follow them for a really, really, really long time. Okay. There are retrospective cohort studies. There are prospective cohort studies. And you can kind of see down there, perspective, looks at collecting information over the course of a period of time and what that does on someone's, ultimately what that does for them in the future. And I think I have that. There we go. Retrospective looks at past exposures and to identify current health outcomes. Dude eating what's he eating? Pizza? Looks like a wedge of cheese for all I know. All right. But what were his or hers past things that they did? These are hard sometimes because this requires remembering. What did you guys eat last week? What did you guys eat last Tuesday night? Exactly. I don't know. I know. I had chicken. Chicken, green beans, and sweet potatoes. I do that every night, or at least right now. So I know what I ate. But most people, if you're asking them to remember what they ate, when they were in their 20s, that's going to be a huge generality. How much did you eat? Oh, I ate pizza. Okay, how much? I don't know. Some days I ate like 15 slices. Some days I ate 2. I don't know. So you can kind of see there is a little bit of limitation there, right? But what does this allow us to do? Look at individuals at that time now, be like, okay, you should probably limit your slices of pizza a day to, maybe 2, maybe 1, because in like 30 years down the road, you're going to have, really bad cholesterol, lipid panel is going to be all screwed up. You're going to be overweight. You're going to have all these health conditions. That's what we can get off of that. Okay. That's what we can suggest an individual do. Some correlations there. Prospective. And looks at diseases that individuals can develop. So we are following this individual over the course of a period of time. The longer the study, the better. The longer the study, the more expensive it is, though, too. And you have to have a large number with these type of groups, right? Just in case people start to die off. Right? So if you only have 20 people and 10 die, that stinks. Okay? But these prospective cohort studies, they try to ID links between disease and what's changed over time. Right? So this, you can kind of see it in real time almost, what in the world is happening and going on with these individuals. So these are really pretty good type of studies, these prospective cohort studies. no matter what we do, retro or prospective, we're looking for correlations. We're looking for relationships. Right? There's a variable. They ate 3,000 calories a day. What does that do to the individual long term? They ate 1,000 calories a day. What did that do for the individual long term? We can look at various different variables. Okay? We can parse out everything that these individuals have done and really try to figure out, has this been a correlation that is positive or negative? Now, if it's positive, it can increase or decrease in the same direction. Okay? So if they're both going up or they're both going down. Right? In the same direction. Okay? It could be a positive correlation in that regard. Those who ate more vegetables saw greater longevity in life. Okay? Those who ate fewer vegetables, that's a positive correlation. Those who ate fewer vegetables didn't live as long, going down. Again, it's a positive correlation for negative health outcomes, would be the way to say it. Negative relationship where one goes one way and one goes the other way. Okay, one increases and one decreases. Again, causation is just really difficult. Really difficult. Very difficult to prove. Okay, there are a billion variables out there, and it's really hard to think of them all and control all of them. Control all of them in a free living individual. Okay, once you get your experiment done, you've done your data, you've done all that summarization, you've written it all up, you publish it, peer review it, and have a critical analysis of your work. Okay. The last part that we don't really care about. And then lastly, research bias here. Again, I made reference that most all scientific journals anymore have at the very end of the article generally a small little summary of any potential research bias that there may be out there. It just, who funded the study generally is kind of what is reported in this. It's not a huge part of a scientific write-up anymore. Well, it never really was to begin with, but it's just 4 or 5 sentences that just kind of explains, this is an experiment that was on this and it was funded by X. Okay, and it was done at this university that has a huge grant with this. So that's all that is. That allows us to have really a proper frame of reference of where the study is. Because a lot of studies done, and I don't even remember when this started being implemented, but a lot of research in the 80s and in the early 90s, you didn't have this. And there's a ton of research coming out that would suggest, that this food or this drink or whatever was healthy for you. And it's funded by Coca-Cola. Well, of course, you're going to, shocking, right? Coca-Cola is going to come out with a study that says Coca-Cola is healthy for you. Okay? Now we have this at the end of research articles where it says who funded the study, if it was funded by anybody, who funded the study, and anybody who worked on the study and any of their potential influences. Okay, the lead person in the study works for the dairy industry, and it's something on milk. Okay? Now, sometimes it doesn't matter. The outcome of the research didn't support, let's just say, milk, for lack of a better one. And so you can really take this with positive attitude, and that it was really scientific. if it comes out that, let's just say milk, and I know I'm picking on milk, but that's okay. They're big boys. There was a positive outcome with consuming milk. Okay, and it was funded by the Dairy Association. Okay, that's still okay. Maybe you go through the research article and you read it and you can see, okay, man, good gosh, they really accounted for all the variables and they really regimented and everything, and it was still positive. That's okay. It's not necessarily a bad thing if you have a potential bias. If you just look at the information and look at the data. Follow up, and follow up again. That's what's been happening since 1900.